Last year, Professor Whittingham, Nobel 2019 Nobel laureate in chemistry, suggested strongly in our interview with him, we should have, the young people have great discoveries. So we should have in the plenary, young and scientists that have achieved so much in their discoveries. So that's why we have here a presentation with a great discovery related to Alzheimer's. Alzheimer's today is one of the most serious sickness around the world, and there is no solution curing it. So this is a fundamental study that has so huge implication in finding a real solution, a real medication in preventing and especially curing Alzheimer's. So the next presentation is from Davis Joseph. It is the discovery of neurobiological mechanism behind 4E-BP2 protein, DEAMIDATION, and its impact in the neurochemistry of the nervous system. So Davis, please.
Thank you very much, Dr. Congoli. Can I be heard? Can you hear me? All right. So thank you very much for attending. Today I present to you my discovery related to the fundamental neurobiological mechanism of protein deamidation in the brain. So I will start off by saying this was solo work. This was work done independently. Without the support of a supervisor, I conceived the theory, I performed the experiments, I conceived the experiments and validated my work. Before we begin, I'd like to make a very quick acknowledgement to my mother, the best mother ever. She was Dr. Megan Debra, PhD in law. I wouldn't be who I am today if it wasn't for her. She sadly passed away a few months ago during my graduate studies. I love her very much and I always will. I will start off by doing a quick background of my research and of my publication. Since I know there are many people outside of my field in this room, distinguished chemists, physicists, biologists, engineers, CEOs of many prestigious companies, I want to make my work as clear and concise as possible for everybody to understand, even though you are not in biochemistry specifically. So I will start off with a flow sheet of life, going into the specific details of the biology that preceded my discovery. We'll start off with the basics, with DNA. DNA is the fundamental code of the human body, containing the genetic information necessary for the development of all the cells in living organisms. And we follow up on DNA with messenger RNA, which carries genetic information. Messenger RNA, what's going on here is that the genetic information is taken from DNA and copied into the messenger RNA, a process known as transcription. The messenger RNA uses molecules called nucleotide molecules to carry the genetic information copied from DNA. As a follow-up on this, we have, this is my subfield, the 5' messenger RNA cap structure. So this structure is conceived to protect the messenger RNA from degradation. It's also made up of the following parts. It has a 7-methylguanosine, a nucleotide molecule which contains no genetic information, but is, however, essential for the messenger RNA's functions. It also contains a nucleotide molecule containing the genetic information, the first of the messenger RNA sequence of its kind. And both inside this cap structure are connected by a triphosphate bridge, which is essentially a linker for both nucleotides. As a follow-up on this, we have this super important protein the EIF4E protein. This is, as you can see right here, this protein is of tremendous importance because the messenger RNA cap structure is connected by intermolecular forces to the eukaryotic initiation factor 4E, EIF4E for short. And this protein was discovered by a great scientist, Dr. Witold Filipovich, in 1976. You can see his paper right here. He started the subfield of translational control. He's the pioneer in this field. And EIF4E, what it does is it directs ribosomes to the messenger RNA 5' cap structure. Now, what are ribosomes? You can see the ribosomes right here. They produce the proteins. Ribosomes are macromolecular machines that copy genetic information from messenger RNA. Ribosomes use this genetic information to produce proteins, and this process is known as translation. So, finally, we get to the proteins. Proteins produce the chemical reactions which allow us to live, as we saw with the very important HIF factors presented by the great Dr. Semenza. Proteins are very complex molecules, such as enzymes, hormones, antibodies, responsible for all the functions and structures of the cells of the living organism. Proteins produce all the chemical reactions which allow us to live, and would otherwise be impossible without them, such as oxidative phosphorylation. Now, finally, we'll also be looking at the proteins I'm studying, the 4EBPs, which stop protein production. So, 4EBPs are very important proteins because 4EBP proteins discovered by Tae-Yan Lin et al. In 1994 attach to EIF4E, a process known as binding. The binding stops the recruitment of ribosomes by EIF4E, a process known as inhibition. As a result, proteins can no longer be produced. So, essentially, you've got two competing factors here. You've got EIF4E, which binds to the 5' mRNA cap structure and directs ribosomes to said structure to produce proteins. And then you have 4EBPs, which bind to EIF4E and stop EIF4E from directing ribosomes to the cap structure. As a consequence, proteins can no longer be produced, like I said. A proper balance of these two competing factors is crucial for the correct functioning of the organism, a concept known as homeostasis. Now, 4EBP2 is the protein I'm specifically studying. This protein is amazing. 4EBP2 is a member of the 4EBP family found in the brain specifically, at much higher levels than any other 4EBP protein. 4EBP2 controls all brain activity because it's responsible for regulating the production of proteins in the brain. 4EBP2, well, all chemical reactions in the brain are regulated in one way or another by 4EBP2 because it's implicated in all neurochemistry, as seen in previous studies. 4EBP2 is found everywhere in the cells of mammals. However, 4EBP2 undergoes a chemical reaction called DEAMIDATION only in the neurons of the nervous system, discovered by Birinosti et al. In 2007 and published in 2010. This chemical reaction stops 4EBP2 from binding to EIF4E, and in this way makes possible the production of proteins again. So, 4EBP2 is not destroyed by this chemical reaction or destabilized by it, as is usually the case, but it can no longer complete its functions because of deamidation. So, this leads to my research focus. Why does 4EBP2 deamidation happen only in neurons? Why nowhere else in the body? So, for 16 years, despite extensive research by the experts in the field, there was no credible explanation as to why this reaction happens specifically in neurons and nowhere else in mammals. So, this is very important because 4EBP2 deamidation significantly alters protein production in the nervous system and maintains the balance of protein production that is required for having a healthy nervous system, homeostasis. Any imbalance in protein production in the nervous system causes neurodegenerative diseases. Discovering what causes 4EBP2 deamidation will allow us to control this balance of protein production and develop effective treatments against neurodegenerative diseases, including memory loss. So this leads to my discovery. My work. On August 31st, 2023, I started work to discover the reason why 4E-BP2 deamidation occurs only in the nervous system. On September 13, 2023, when I was only 23 years old, I achieved this goal. And I'm going to present to you my discovery right now. So, when I got into this research, my hypothesis was the following. 4E-BP2 deamidation happens only in neurons because of the axons and their unique properties. What are axons? They are long extensions of the neurons which conduct electrical impulses that are crucial to nervous system activity. I used immunoblotting experiments, also known as Western blots. Western blots separate proteins based on their molecular weight. Western blots can detect 4E-BP2 specifically thanks to antibodies which target the protein. 4E-BP2 extracted from the nervous system appears as 3 bands on Western blots. And this is what it looks like. This is 4E-BP2 on a Western blot. It appears as 3 bands. The top band represents complete deamidation. The middle band represents partial deamidation. And the bottom band at 4E-BP2 represents no deamidation. And here you can see GAP-DH. GAP-DH is the loading control. It's used to make sure that the same amount of protein is used in all samples. This is what I did. And I also invented a new method of calculating protein deamidation, which was crucial to all my research, called the deamidation ratio. You divide the complete deamidation band intensity, the intensity of the band at the top, by the no deamidation band intensity, the intensity of the band at the bottom. And then you get the portion of 4E-BP2 proteins which are deamidated in the specific sample that you're analyzing. This was my creation. I used mouse models. Why? Well, all experiments were conducted firstly on 2-month-old wild-type male mice models. Wild type means that the mice were non-mutated. They're mice you can find in nature, but preserved in the lab. Mice are used as models because their nervous systems are representative of human nervous systems. Also, mice of the same age and sex were used in order to avoid data variability due to outside variables related to age and sex specifically. Now, this leads to my experimentation on the central nervous system. What did I do? Well, the first goal of my first experiment was to compare 4E-BP2 deamidation in the 2 parts of the neurons of the brain. A, the axons, B, the cell bodies, also known as the soma. The optic nerve and the retina were chosen for analysis because they are large extensions of the diencephalon, a division of the brain, and are isolatable via dissection. Their physical properties are identical to the rest of the neurons in the brain. I isolated the axons in the optic nerve and the cell bodies in the retina in order to determine if 4E-BP2 deamidation is happening mainly in the axons. This was the first time ever that the optic nerve and the retina, were dissected in order to detect 4E-BP2 deamidation. Never been done before. There was a second goal in my first experiment. It was to compare 4E-BP2 deamidation in the whole brain, which is a mixture of axons and cell bodies, with the individually isolated axons in the optic nerve and cell bodies in the retina. This comparison shed light on where exactly 4E-BP2 deamidation is originating from. These are my results. Based on a 4E-BP2 Western blot of the whole brain, the optic nerve, and the retinal ganglia of mice, I found the following. Deamidation is significantly higher in the optic nerve compared to its retinal ganglia. There is no significant difference between the deamidation of the optic nerve and the whole brain. There is, however, a significant difference in deamidation between the whole brain and the retinal ganglia. Conclusion: deamidation in the central nervous system is coming from the axons, as my hypothesis predicted. Which leads to my experiments on the peripheral nervous system. My experimentation here was the following. the goal of the second experiment was to compare 4-EBP2 deamidation in the two parts of the neurons of the periphery of the body, just as in the brain. A, the axons, and B, the cell bodies, the soma. The sciatic nerve and the dorsal root ganglia, DRG for short, were chosen because the sciatic nerve contains the longest axons in the body. The DRG contains the cell bodies of the sciatic nerve. This nerve is far from the brain, which is important because I wanted to see if what I found in the brain was systemic to the entire body. So I chose the nerves that were as far away from the brain as possible. I isolated the axons in the sciatic nerve and the cell bodies in the DRG in order to determine if 4-EBP2 deamidation is happening mainly in the axons, just like I did in the brain. This was the first time ever that the sciatic nerve was dissected in order to detect 4-EBP2 deamidation. Again, never been done before. These are my results. Based on a 4-EBP2 Western blot of the sciatic nerve and the dorsal root ganglia, I found the following. 4-EBP2 deamidation mainly occurs in the axons of the peripheral nervous system. 4-EBP2 deamidation is significantly higher in the axons of the sciatic nerve compared to its soma located in the dorsal root ganglia. The findings in the PNS, peripheral nervous system, corroborate, therefore, the findings in the central nervous system, or CNS for short. Conclusion, deamidation in the peripheral nervous system is coming from the axons, both in the central nervous system and in the peripheral nervous system. Then, I did a comparative study. The goal here was to do a comparison of the rates of deamidation made between the whole brain, where axons are mostly myelinated, myelination means isolation, and the sciatic nerve, where axons are mostly unmyelinated. Unmyelinated meaning not isolated, in order to determine the effect of myelination on deamidation. My conclusion based on the results here was the following. Deamidated 4-EBP2 is more enriched in the myelinated axons of the whole brain compared to the mostly unmyelinated axons of the sciatic nerve. This led me to my theory, which I established based on these results. I had three conclusions based on my results. Deamidation in the central nervous system is coming from the axons. 1. 2. Deamidation in the peripheral nervous system is also coming from the axons. 3. Deamidated 4-EBP2 is more enriched in the myelinated axons of the whole brain compared to the mostly unmyelinated axons of the sciatic nerve. I wanted to study the science behind these experimental results. So what I did here was I conceived an organic chemistry deamidation flow sheet consisting of 5 intermediaries which I invented to better understand the reaction. We don't need to go into the specific details of the organic chemistry behind 4-EBP2 in this context. All we need to know here, what's important, is that deamidation is a spontaneous chemical reaction which consists of an asparagine in 4-EBP2, which contains nitrogen, being converted into an aspartate molecule in 6 steps. The nitrogen is converted into an oxygen. That's what we need to know from this slide. So, 4-EBP2 deamidation occurs in asparagines 99 and 102, based on previous studies. Asparagine goes to aspartate, as said previously. The deamidation half-life is an important concept here because it is the time required for 50% of the asparagines to be converted into aspartates, based on Robinson's algorithms. The protein half-life is the time required for 50% of proteins to be degraded. I concluded that the deamidation half-life of asparagines 99 and 102 is reached 18 days after birth, because this is consistently the time that is necessary to observe 4-EBP2 deamidation significantly. And based on this research and my experimental results, I concluded that 4-EBP2 must have a protein half-life greater than 18 days for deamidation to occur. What do I mean by this? The reaction takes 18 days to occur. If the protein lives only for 14 days, you're not going to see deamidation. But if the protein is found somewhere in the body where it lives, say, 25 days, then you're going to see the reaction. So I thought, okay, so it's happening specifically in neurons. That means it's living longer in neurons. But what is causing this increase in life in neurons? Well, based on my experimental results, I had the answer. Axons are found to increase protein half-life. And I did research on other proteins to confirm this. The SOD1 protein has a half-life of 100 hours in kidney cells, based on a previous study. Whereas half-lives of the same protein, when found in the axons, are over 1 year. The protein only lives for 4 days in kidney cells, but in axons it lives for over 1 year, which is amazing. 4-EBP proteins have a half-life of only 20 hours outside of neurons, which is way below the threshold of 18 days previously established. 4-EBP proteins are also found to have a significantly longer half-life in neurons versus kidney cells. Then, my experimental conclusions 1 and 2 confirm that deamidation is specifically happening in axons, both in the central nervous system and the peripheral nervous system. Based on this conclusion and my results, I concluded that 4-EBP2 half-life is significantly increased by the axons in the neurons, which leads to deamidation occurring specifically in neurons. You need a longer life for the protein for this chemical reaction to occur. And it is specifically the axons, based on my results, which are causing this longer life. I also concluded that 4-EBP2 deamidation is controlled by the axons, based on my results, which led me to my next question. Why? Why are the axons responsible for increasing the life of proteins? Well, I had the answer based on my results again, and based on the research I did. I looked into red blood cells to get this answer, because everything in the body is connected. Proteasomes degrade proteins. Proteins in proteasome-poor environments, such as red blood cells, also known as erythrocytes, have significantly longer protein half-lives. Axons are also found to be proteasome-poor compared to the soma, the cell bodies, due to limited ribosome biogenesis. Proteasomal activity has been found to be higher in neurons without myelin, because they're not isolated. They're consistently in contact with the extracellular environment. There's protein exchange, etc., etc. My experimental conclusion confirmed what? Exactly this. That deamidated 4-EBP2 is more enriched in the myelinated axons, where there's less proteasomes, than in the unmyelinated axons, where there's more proteasome. So, based on my experimental conclusion, I concluded that the proteasome-poor environment in the axons is what makes 4-EBP2's half-life longer and deamidation thus possible. I also concluded that 4-EBP2 deamidation is maintained at different rates per neuron cell type throughout the organism, depending on the function of said neuron. More corroboration of my findings. Mr. Richie Chuang, he corroborated my experimental results by previously detecting 4-EBP2 deamidation specifically in skeletal muscles. This corroborates my theory. Why? Skeletal muscles are the part of the body that have a very, very high density of axon terminals found in the neuromuscular junctions. And this is the only place besides the nervous system where we can detect 4-EBP2 deamidation. This leads me to the principle I founded. Due to their proteasome-poor environment, axons increase the protein half-life, which becomes more significant than the deamidation half-life of asparagines, creating neuron-specific deamidation. The biological clock is now observed. My contribution fundamentally changes our understanding of protein production by demonstrating that the axon is a regulator of protein production, of translation in itself. The axon causes deamidation in the 4-EBP2 protein, and by doing so, stops 4-EBP2 from inhibiting protein production in the nervous system. The axon is a translation enabler, based on my results. And this is my contribution to the flow sheet of life. Axons create 40pp2 deamidation, which allows protein production to progress. Next steps. Why are my findings scientifically important? 40pp2 is fundamentally linked to memory. Understanding the cause of 40pp2 deamidation leads one step closer to understanding the inner machinations of human consciousness, because 40pp2 deamidation is a crucial regulator of protein production in the brain. The absence of deamidated 40pp2 in the brain leads to the development of Alzheimer's disease. Deamidation is related to aggregation and amyloid plaque formation, a process which is the root cause of memory loss in Alzheimer's. 40pp2 is indispensable to the survival of all mammals, because 40pp2 deamidation has been conserved in all mammals for 90 million years. The protein sequence in probe 40pp2 that causes deamidation has been conserved in every single mammal. Humans, chimpanzees, zebras, rats, mice, etc. And there's a reason for this. It's essential to the survival of the mammalian brain, and therefore crucial to all mammalian brains. Financially, the findings are important because the Alzheimer's Association predicts the finding of a drug treatment, not a cure, just a treatment for memory loss, could save families and the government $935 billion over 10 years in the United States alone. Forbes 2015. Next publications. This work is a precursor of 2 other subsequent discoveries which I made after finishing this publication. I discovered why 40pp2 is the dominant member of the 40pp protein family in the brain, and I discovered and prepared a new and effective treatment for memory loss. Both discoveries are also on their way to publication. They're on the verge of publication. And therefore, thank you very much. Efharisto. This is my work.
It's amazing, amazing, amazing. Congratulations, Davis. Your mother from heaven is proud of you. I'm proud of you. And I think in the audience, people feel the same. He developed this theory when it was a lot of stress because of loss of his mother. Thank you again. It's this we hope, and I'm sure it leads in a cure, not only treatment, but a cure of Alzheimer's disease. So I am an axon. So the theory is basically axon. This is the key point related to Alzheimer's disease. So again, congratulations. Congratulations. And another applause. We have really a big discovery here. Yeah.